Good evening, everyone. In our last class, we were discussing about this luminometry, okay, and we have seen that when we work with the luminescence or or what you call when we work with the luminescence and we want to find out the luminescence, the lamp and the excitation optics, they are not needed. Okay, in fact, they have photomultiplier tubes in place which are placed very close to the sample so that we can reduce the loss of the photons. Okay, so we have looked into the aspects, how we are getting this thing. So we had this sample chamber, then we had the photomultiplier tube, then from here there are amplifier detectors and then recorders and all these things. In some cases we have also discussed that there may be some monochromator or the filters. So this monochromator or filter, it will allow you to filter out a specific wavelength radiations or the visible lights to pass through, which can reach the PMT detector. Okay, and we have also discussed that the measurement of the signal is expressed in the relative light units. Okay, then we have discussed about tube illuminators and microplate illuminators and multimode microplate readers also. So then we discussed about the bioluminescence. We have talked that luciferin, which is a biological compound, it is being metabolized into this oxyluciferin by the process of oxidation, and during that process the bioluminescence is being generated. And we have seen the this reaction, this oxidation of the luciferin, it occurs in the presence of the enzyme luciferase, which acts as a catalyst. Okay, and we have seen at a wavelength of 440 to 479 nanometer, we have this bioluminescence. Okay, and this is the chemical reaction, okay, which usually takes place in most of the cases. And then we have talked about BRET, and we have seen that BRET is basically used to understand the interactions between two proteins. Okay, and if the distance between the two proteins is less than 10 nanometer, then this there is a transfer of the energy, okay, from this R luciferase uh, and to the fluorescent protein EYFP, enhanced yellow fluorescent protein. Okay. So we also discussed about the different methods of these breads, okay, and what are their donor emission and the acceptor emission, and accordingly we have seen that this broader this donor emission and the acceptor emission is there, so better it is to capture the signal, a specific wavelength signal. Okay. So we have briefly discussed about all these techniques, okay, and then we discussed about FRET, which is not a part of the bioluminescence or the luminometer, but it is an analog of BRET, and hence we have discussed how FRET is different from the BRET. Okay. So today we are supposed to start with this reporter gene assays. Okay. So let's see into the over here what happens. So what does reporter gene assays do? It is an invaluable tool for us which helps us to study the gene expression. Okay. Now, when we talk about a gene it basically consists of two main functional units. Okay. The first consists of a so-called coding DNA sequence that provides information about the protein being produced. Second, a specific promoter sequence linked to the coding that regulates the transcription of the gene. The promoter serves to activate or suppress the expression of the gene as required. Okay, so basically this promoter sequence, it is, we call it promoter, and this coding DNA sequence which generates or which provides information about the proteins that will be produced, it is regarded as the reporter. Okay. We will see later on how it functions with the help of a diagram. The main purpose of the reporter gene assay is to investigate the promoter of a gene of interest. This is an important thing. So we are not interested in this reporter part, okay, which is coding for the protein, okay. In fact, we are interested in this promoter sequence, okay, which is responsible to either promote this reporter gene or suppress the reporter gene. In other words, this promoter sequence, it helps us uh, in the expression of the reporter genes, so that either it can produce a high level of proteins or low level of protein, or an average amount of protein. That means how much protein will be expressed by this reporter gene will be controlled by this promoter sequence. Okay. So, as we were discussing, the main purpose of the reporter gene assay is to investigate the promoter of a gene of interest, that is, the regulation of its expression. This can be done by linking the promoter of interest to an easily detectable gene, okay, such as the gene for firefly luciferase, which catalyzes a reaction that produces light. Okay. Usually the cells are then exposed to different factors or conditions, or changes can be made in the order of the reporter, the effect of which can be easily tracked by measuring the changes in the light emission. Okay. So what it says, it says, if I go in the next slide, so usually what happens is that you have talked about, you have heard about this uh, what do you call genetic this what do you call genetically modified plants, or genetically modified this thing, your microbes. So these are basically used over there. Okay, so in the genetically modified systems, what we do usually, we have a plasmid, especially in the case of the E. coli in the microbes, which is being used as the test microorganism. So what happens? We have a plasmid, and in this plasmid, we have a sequence which is regarded as the promoter. So, okay, so here promoter of interest, it is in green color, so I have marked it in green. Okay, then we also have another gene sequence which is just nearby this promoter, and it is the gene of interest, for which you want to find out uh, uh, whether this promoter sequence, it will be able to activate this region, this gene of interest, so that it produces the desired proteins. Okay. Now you want to study about this thing. Now what you will do, you will take this plasmid and you will transfect it into the E. coli. Okay, so say this is the E. coli, so we will be transfecting it within the E. coli. So it will go within the E. coli and it will start producing the proteins. But how much proteins will be produced? It is being governed by this promoter. Okay, but you have developed this plasmid on your own, so you don't know how it will behave. So you need to find out how this plasmid will act. In this case, what you do is that instead of using this original gene of interest, what we can do is that we can take the plasmid, we take the reported promoter gene, and we take the reporter gene. Now reporter gene, as the name suggests, it will tell you that it is present or not. So it is just reporting to you. Okay. So what it says is that what it does is that when you induce or transfer this into the E. coli, this reporter gene, what it will be doing, it will be synthesizing the luciferase enzyme. If you see over here, we have talked about the such as the gene for the firefly luciferase. So what we can do, this blue part, the reporter gene, it will be basically the gene sequence which is responsible for the generation of the firefly luciferase enzyme. So it will produce this reporter gene, it will produce firefly luciferase. Okay, or LUC, I think we have named it as LUC. Let me check again. So that bread 3, yeah, bread 3 over here, we are having this. Okay, this firefly, so we're naming it as firefly. So in short, we will say firefly luciferase. Okay, so this reporter gene, it will generate the luciferase, firefly luciferase enzyme, and this promoter will now, whether it is activated or it is suppressed, whatever the thing it is happening over here, we will now be able to find out the thing by estimating what is happening to the luciferase enzyme. Okay, so what it will do, when we transfect it within the E. coli, so then we put this E. coli in different conditions. So when we put this E. coli in different conditions, then we find out the conditions at different conditions, how this promoter is behaving, and what is the quantum of the luciferase enzyme which is being produced over there. Okay, so what it will do, depending upon, suppose you have this sequence, this is the promoter and this is the reporter. Okay, now this reporter, it will generate the enzymes. Okay, firefly luciferase, I will name those as FL, but this cannot generate the protein without getting the command from this promoter system. So it will get if get an instruction from the promoter to generate this thing. Okay, suppose you put the E. coli in a what do you call highly saline condition. Then say under that condition there are only two luciferase. These are just for the sake of making you understand. So it will produce a lesser number of the firefly luciferase enzyme. Suppose you keep it in a say what do you call mm, in a conducive environment, say in a broth, in a nutrient agar broth, not a nutrient broth, not nutrient agar broth, nutrient broth, and then you incubate it at 37 degree. So E. coli will get sufficient nutrient over there to grow. So maybe at that time this promoter may be activated. So that is another condition. Okay. So under that suitable condition, it will send the signal to the reporter gene to produce more number of FL. Okay. Now when you are producing the more number of FL, the intensity of the bioluminescence from the E. coli will increase. Okay, and this is what we do in this reporter gene assays. Okay, so usually what you have the promoter of interest and gene of interest, but this gene of interest, it produces a separate protein which is altogether different from what you will be testing in the reporter gene assay. Okay, suppose this gene of interest, it is producing say any protein A. Okay, and it has nothing to do with the firefly luciferase, but you want this protein to be expressed say within the human cells, or say you may say that you want insulin to be produced within the human cells over here. You can do that. Okay, uh, or in microbes you may be interested to express some other say antimicrobial peptides, so that is also possible. So it has nothing to do with the assays. What you are interested in this, in the promoter of the interest, promoter of interest, because ultimately this promoter of interest, it will be controlling this gene of interest to produce these proteins. Okay, in the second case you have a promoter, say in the firefly you have this promoter, and then you have the reporter gene in the firefly which will produce this reporter protein. In this case it is firefly luciferase, and this firefly luciferase, it will catalyze some reaction to generate bioluminescent what you call radiations. Okay, and which can be detected using the luminometer. Okay, now here if you see, you do not know anything about this promoter, but you are interested over here in the reporter gene. So in the first case you will take the promoter of interest. From the second case you will take the reporter gene. Then in the third part you will ligate this promoter of interest and the reporter gene, okay, within a plasmid, which will then be transfected within the either the microbes or the human cell, whatever you say. Now you subject that cell to different conditions. Okay, and see how much bioluminescence, in this case we are talking about firefly luciferase, how much bioluminescence are being generated. Higher the bioluminescence, intensity of the bioluminescence, the higher is the activity of the promoter of interest. Okay. If there is a lower bioluminescence, then the activity will be lower. Okay. So this is what you will be getting. Okay. So am I clear up to this point?
Yes, sir.
Okay, good. Then there was some problem with this uh, uh, single reporter gene assays, where we are having only one promoter of interest and one reporter gene, and we are having there were some issues because uh, what is the attenuation of the signal, you don't know. Then the number of, if the number of your these microbial cells, or the or say no, in general the cells, if you increase the number of the cells, then it might so happen that the cells are absorbing some luminescence, the other cells. Okay, so that will give you an erroneous result, so you do not have any information about that. So under that condition, what to do? So the researchers have come out with an unique way. What is the way? So what they are saying is that apart from the experimental reporter which will generate this firefly luciferase, we will put one control reporter also which will generate renilla luciferase enzyme. Okay, control reporter which will generate renilla luciferase. Usually the ratio of this is either 1 is to 1 or 2 is to 1. So this is experimental reporter and control reporter. Okay, so if you go back, renilla luciferase, see, it is having emission at 480, and firefly luciferase, it is having emission at 565. So there is a wide gap. Okay, so you can use monochromator to isolate this bioluminescence, and you can use another monochromator to isolate this bioluminescence. Okay, so you can individually measure the information from here. Okay, now once you have got that, then you look into the ratio pattern. So whether it is in the 2 is to 1, so so you know the ratio. Okay, so say you have x, x for say experimental reporter, and then for the control reporter you have say 1. Okay, now it is equal to 2 is to 1. So if you get 1 unit of bioluminescence, that means for x you should get 2 amount of bioluminescence. Okay, so you can verify the results, and from here uh, you can find out whether there are other noise components or not. So this will allow you to have a more accurate result, because you have a control reporter and you know you can cross check whether it is in the 2 is to 1 ratio. You have put it in the 2 is to 1 ratio, and then you can look into the luminescence pattern. So right, am I clear?
Yes, sir.
What about others? So what I am saying, you have experimental reporter and control reporter. You transfect these reporters in the ratio of either 2:1 or 1:1. Now, individually you measure this one, individually you measure this one. Okay. Now, what you can do, you can go for this validation. Okay. So, suppose you have got for the experimental one, x is to say 0.5, which is equal to say in the first case it is 2:1. So, you should get the luminescence of this one as 1. Okay. So in this way you can validate your result. So, and if there are any problem over there, you can look into that thing. Okay. Now ATP measurement. So ATP determination can be used as an indicator of viable cell numbers. Okay. ATP participates in the oxidation of the luciferin by the enzyme firefly luciferase, a reaction that produces light. The quantity of light produced is proportional to the quantity of luciferin oxidized. This reaction is very popular in reporter gene assays where ATP and luciferin are in excess. And the quantity of light produced is limited by the expression of the luciferase, the reporter. However, the same reaction can be used to quantify ATP if both luciferin and luciferase are in excess. Okay. So here what is happening? The enzyme firefly luciferase, it is being synthesized, and ATP and luciferin are present within the cells, and they are available in what you call in sufficient quantity. So it all depends on the firefly luciferase which will be generated, and the reaction is limited by the synthesis of this firefly luciferase. Okay. In the case of ATP, what will happen is that herein both luciferin and luciferase, they are kept in excess, and then we use the same thing for the for quantifying the ATP. Okay. So what would happen? The same reaction, if you look over here as an indicator of viable cell numbers, if there are larger number of cells, if they are rapidly multiplying, you will be having larger ATPs, larger amount of ATPs. So the more the ATPs will be produced, the more the ATPs will be there, the more will be the luciferin, the more will be the bioluminescence. Okay. So if there is a small number of cells, viable cells, then the amount of ATP which will be available, they will be lower, and if it is that is lower, then bioluminescence will be lower. Okay. So in this way you can correlate the viable cell numbers and the bioluminescence, and from there you can find out you can relatively find out whether a material is toxic to a substance or not. Okay. So suppose you have 100 number of cells over here. So you have measured the bioluminescence, ATP, you have done the ATP measurement. Then you add a foreign substance A. If it is that is toxic, it will kill many of the cells. Only few cells will be there. So the bioluminescence will be lower. Okay. In other applications, you can go for hygiene monitoring. So as we said, ATP is present in all living cells and is therefore an indicator of biological contamination. Okay, that means what you are doing, suppose in your area you are having this supply, water supply. Okay, now whether this water supply which is being delivered to your house, it is contaminated or not, you can definitely check. So how will you check? You will take the sample of the water. The ATPs, they will be present in the living cells. You go for this luciferin, this thing, and from there you will be able to uh, see the bioluminescence whether it is increasing or decreasing, and from there you can sorry, if there is any presence of bioluminescence in this hygiene monitoring, if there is any uh, bioluminescence, then that means that water is contaminated with some living cells. Okay, then we can have immunoassay. In the immunoassay, I'm just giving an example. Say you have the antigen which have been immobilized onto a surface, then you have antibodies which is attached with say luciferase enzyme, and then you put it over here within the well. If they bind, it will be looking something like that, right? Now if you wash it off, if they are binding, then they will not be removed. Okay, then you add the this other components, the ATP and the luciferin, then it will generate the bioluminescence. So in that way you can go for the immunoassay. So this is antigen. Then you can have this sandwich assay also, where you have the antibody, then you put antigen, and then again you put another counter antibody with the luciferase enzyme. So in that way also you can get it. Okay, so in this way you can go for this immunoassay analysis. Okay, then we can look into the caspase as activity or caspase assay. Why it is important? Monitoring of the activity of caspases, a group of cysteine aspartic acid peptidases, is a key method in apoptosis research. Okay, so what happens over here is that this enzyme, caspase 3 enzyme, okay, in caspase, this caspase 3 enzyme, when it is activated, there are several ways, there are specifically two ways. I'm not going into the details of that one. So basically, via those two ways, if you activate the caspase 3 enzyme, then it starts the process of self amplification, and simultaneously it goes and specifically kills the cells in a controlled manner. Okay, so if you want to see whether any condition it is activating this caspase 3 or not, how will you find out? Again, you have the petri plates, you have the cells, then you have the media over here. You add whatever the components you want to add, say component A, which is an activator of the caspase 3. When you give an external stimulus, or you give external chemical or an external stimulus, and you want to say whether the caspase 3 is activated or not, then what you do, the ATPs are present within these cells. Again, this firefly luciferase and this luciferin, if you add into the media, then if there is an higher bioluminescence, that means it is not affecting the cells. However, if there is a decrease in the bioluminescence activity, that means somehow the caspase 3 has been activated and it is resulting in the killing of the cells. Okay, so in this way you can find out the caspase activity. Then we have kinase assays. In the kinase assays, as the name suggests, it is associated with some kinetics. Okay, and if there is any biological kinetics going on within the body, then there will be an increased consumption of ATPs, right? There will be an increased consumption of the ATPs. If there is an increased consumption of ATPs, then what will happen? The bioluminescence will be reduced. Okay, so these kinase assays, after giving some stimuluses and all these things, we find out whether the kinase activity it is increasing or decreasing. Okay, the kinases are modifying the activity of specific proteins and are extensively used to transmit signals and control complex processes in the cells. Their enormous diversity and their role in signal transduction make them attractive targets for research and drug design. Okay, so we can find out. Suppose if you find out if you have a drug and you want to find out whether the kinase activity is decreasing or not, you just go for this test, this Luminometry test, and you will be able to find out uh, whether the kinase activity is reducing or not. Okay. Then similar cellular luminescence, so which happens due to the reactive oxygen species. Okay, the cellular chemiluminescence is the production of light after chemical reaction of activated cells like neutrophils, granulocytes, monocytes, or macrophages when phagocytosis is activated as the first immune response. Okay, when phagocytosis is activated as the first immune response in order to protect the organism from the invaders. After the ingestion of the external object, reactive oxygen species such as H2O2, OH negative, O2 minus are produced, resulting in luminescence at a very low intensity. Okay. To enhance the chemiluminescence effect, substrates like luminol and lucigenin are used. Okay, right? So if you see the bioluminescence over here, it is slightly different than whatever we have discussed till now, which uses either ATP or firefly luciferase for analyzing the uh, for generating the bioluminescence. In this case, due to the our body's defensive mechanism, that is the phagocytosis, the defensive mechanism itself produces some reactive oxygen species like H2O2, OH hydronium ion, and O2 ions, O2 minus ions, which are reactive species, and they are luminescent. Okay, so we measure the activity of this. Okay, and usually they have very low intensity luminescence. To increase the luminescence, luminol or lucigenin are used. Then lastly, we have the water toxicity testing. So the use of intact luminous photobacteria like Vibrio fischeri and Photobacterium leognathi. Okay, so if I have done a mistake over here, so these are scientific names, so they should be either underlined or they should be italic. So please correct this at your end. Or dinoflagellates for toxicity assessment has some clear advantages that has been scientifically validated. These microorganisms are self-maintained luminescent units that under proper conditions emit high and steady levels of luminescence. The intensity of its luminescence is strongly affected by a wide range of contaminants, including pesticides, herbicides, fluorinated hydrocarbons, heavy metals, agents that affect the cell's integrity, membrane function, or cell respiration, or that have an impact on the rate of protein or lipid synthesis. Okay. So what it says basically, this long story in short, uh, suppose you have a water, you add this luminous photobacteria, you don't have to do anything, you just add this luminous photobacteria. If the contaminants like the pesticides, the herbicides, the chlorinated hydrocarbons, heavy metals, and all these things are present, which are toxic, they will be toxic to these microbes too. Okay. Now, these microbes, they are self-maintained luminescent units. Okay. So higher the higher microbe content means higher bioluminescence. However, if the water is toxic, then the microbial content will come down. Okay. So if that is happening, that means your bioluminescence will also be lower, and in this way we can quickly analyze whether a given water is toxic or not, or in other words, in whether in the given water we have the toxic contaminants like pesticides, herbicides, and other chemical compounds. Okay. Not only this, we can also look into the other solvents also, and we can see whether there are any toxic components within the solvents like fruit juices and all these things. So we can definitely check it. Okay. So this is all about your luminometry. So if you have any questions, please let me know.
Sir, here you mentioned about the photobacteria, so we can't use the fungi or algae.
Yeah, if they are luminescent, you can definitely use it. But these are the you can design, definitely you can design your own experiments. That's not a problem. But usually this is the general practice.
Using genetic engineering, we can modify.
Anyone else who has some question? Okay, then we will stop here today.